In this video, we'll talk about the protein buffering system and how it helps in regulating the acids and bases in the human body. Proteins made up of smaller units called amino acids plays a very important role in many bodily functions. Some of them include muscle and skin repair, enzyme and hormone formation like pancreatic juice, and immunity and buffering system capacities. Since this video is about the protein buffering system, they act as buffers by virtue of accepting or donating hydrogen ions whenever the environment becomes acidic or basic. The protein buffering system are of two types. The first one is the extracellular buffer and the second one is the intracellular buffer. First, the extracellular buffer. The extracellular buffer are present as plasma protein in blood and the most abundant of all being albumin. In order to understand the mechanism of the extracellular buffer, we must understand that amino acids in the protein molecule consist of two things. One is the amino basic group, which accepts the hydrogen ion, and the carboxyl group, which acts as an acidic group which donates the hydrogen ion. For example, when the environment becomes acidic, a lot of hydrogen ions are formed. Then the amino group, which consists of two hydrogen ion in the amino acid, will accept the hydrogen ion from the environment, and this will be converted to the ammonia group, which is a very weak acid. Conversely, if bases in the form of hydroxyl ion are added to the body, then the hydrogen ion of the carboxyl group will donate to the hydroxyl ion, and this will form water, thus neutralizing the change in the pH. However, there is a disadvantage of the extracellular protein buffer. The disadvantage being that it has got a very limited buffering capacity. This is because the plasma proteins in our body are present in a very low concentration. Next, we come to the intracellular buffer. The intracellular buffer constitutes about 60 to 70% of all buffering capacity of proteins, a very huge amount. The intracellular buffer is present in the muscle cells. It is also present in the liver cells, but the most dominant is the hemoglobin in RBC. Here are some figures of the intracellular buffer. As we know, the concentration of hemoglobin is around 15 gram per deciliter in blood, whereas the concentration of plasma proteins in blood is around 7 gram per deciliter. That means that hemoglobin concentration is 2 times more than the concentration of plasma proteins, and the hemoglobin buffering capacity is 6 times more effective than the plasma protein, making a very strong buffer. The mechanism of hemoglobin buffer is as follows. In the tissues, carbon dioxide is formed from cellular metabolism, and this carbon dioxide enters the plasma, and from the plasma it gets access into the RBCs of the cell, where oxygen is bound to hemoglobin, forming oxyhemoglobin. Now this carbon dioxide then reacts with water, forming the weak acid called the carbonic acid. Now in the RBC, the carbonic acid formed from the interaction between carbon dioxide and water dissociates into hydrogen ion and a bicarbonate. Now this hydrogen ion will cause a low pH, which will then have this hydrogen ion will bind with the oxygen hemoglobin, forming deoxygen hemoglobin plus oxygen. Now this oxygen then enters the plasma, and from the plasma it gets into the tissues where it is utilized for metabolism. Now the bicarbonate, which now enters from the RBC into plasma, and because there is a negative charge loss, chloride will enter from the plasma into the RBC, and this is called the chloride shift. Now what happens to the lungs? The lungs are filled up with oxygen, which is derived from the atmosphere. Now the RBCs which arrives in the lungs, the oxygen is present in a deoxygenated form. So it is in the form of deoxygen hemoglobin. Now the oxygen will bind with the deoxygen hemoglobin, forming what we call the oxyhemoglobin along with the hydrogen ion, which will reduce the pH of the RBC. Lungs have already given the oxygen, and the RBCs, as we already said, the oxyhemoglobin along with the hydrogen ion is formed. The hydrogen ion, because of the loss of low pH, will bind with the bicarbonate, and this bicarbonate interaction will cause again the formation of the carbonic acid. This carbonic acid then dissociates into carbon dioxide and water. The carbon dioxide so formed will be then expelled out from the lungs, and this is how the hemoglobin acts as a buffer in both the lungs and the tissues. In the next video, we'll talk about the phosphate buffer system. Please give a thumbs up if you like my video, and please subscribe to my channel to get new updates about medical topics.